So company's called Accelerated Biosciences, and we are all about the human trophoblast stem cell platform. How do I go forward? There you go. We are a pure stem cell platform company. We're Delaware C Corp, formed in 2013 and based in Philadelphia. We have complete FTO over the human trophoblast stem cells, we call TSCs, and our strategy is to provide the GMP cells with a full regulatory support package to help you accelerate your therapies to market. The biggest problems we have today are, ethical issues with cells, inconsistent cell source, scalability versus immunogenicity, unclear FTO or CMC documentation issues. We can handle all of that. We solve all of those issues. Plus, our cells are pluripotent. They're ethically sourced, scalable, immune privileged naturally. They're pathogen-free, and they have high chromosomal stability, and we have a large IP estate to protect it. What you see here is a continuum of fetal development, and our cells, TSCs, are sourced from 4 to 8 weeks post-fertilization from ectopic pregnancies, and our cells are mortal, but they're pluripotent. On the left side here, you'll see a blastocyst of 5 days, and our cells are from the trophectoderm lineage, so pre-placental lineage. And in an ectopic pregnancy, what happens is the embryo is traveling down a fallopian tube, and it gets stuck. And in about 4 to 8 weeks, the mother will feel a sharp pain in an abdomen and go to the ER and get scanned. The doctor will say, "Oops, you have a tubal ectopic pregnancy." And they'll schedule an emergency salpingectomy. And they have to do this because if the ectopic mass is not taken out, it might burst the tube, and the mother may die of internal bleeding. It's actually the highest cause of death in first trimester pregnancies. And this occurs about 2% of all pregnancies worldwide. In salpingectomy, what they do is they ablate both sides of the tube, and they take the mass out, and they discard it because the fetus is already non-viable. Because the blastocyst that you see on the left needs to implant normally by day 7 or day 8, but you don't discover the ectopics until about 4 to 8 weeks. So the embryo never had a chance to implant and draw nutrients from the mother to feed the fetus. So it's very understood that the fetus is not viable. And so what we do is we stop the standard of care at the point of removal, and we take the chorionic villus tissue, and we isolate that for our stem cells. So these cells are completely ethical. We're able to turn our cells into many different germ layers, all 3 germ layers: mesoderm, ectoderm, endoderm, NK, cartilage, bone, fat, and kidney for mesoderm, neuron for ectoderm, and pancreas and liver for endoderm. And we've also analyzed the secretome, and we turned our cells into an iPSC, which I'll tell you about in a little bit. We're able to optimize our cells to go out to 85 population doublings. So at 100 million cells per dose, that's 8 times 10 to the 17 doses. That's actually more cells than you have in your body. So this platform will allow you to support an entire product cycle without any problems. And that is the significance of that is to dramatically decrease your cost per dose. We've also put our cells into 5 different animal studies, and we see these human cells survive in immune-competent rats. They home and regenerate the affected organs. We don't see any teratoma or tumor formation, and we get all the behavioral improvements. And the key factor here is the HLA-G. So our cells express HLA-G, and it's the main function, the main expression that prevents the mother's immune system from attacking the embryo. That's why today you can have a surrogate baby. And when we differentiate our cells to neuroprogenitors as well as pancreatic progenitors, you get all the right markers, and the HLA-G sticks. So that makes it ideal for any therapeutics. We tested our karyotype up to 72 population doublings, as you can see here, extremely stable. Some of the mothers that come in, when we test their blood, they may have HBV or CMV. And what we did was we took these cells and sent them to IDEXX for the H-Impact-1 study to test for 25 human pathogens, and the cells come back completely negative. That means there's some defensive mechanism at the fetal-maternal interface that we don't yet currently understand. But that makes these cells a super clean starting point for any cell therapies. We do have to still throw the cells away, so no worries about that. We're able to take these cells and drive them towards an iPSC using a non-integrating mRNA reprogramming method. Then we took a standard kit and drove them towards an NK cell and did a cytotoxicity study, and they kill like gangbusters. So, and we believe the reasoning is because with most other iPSCs, the sources are normally from adult cells, and our cells have never gone through term. They were incubated in the fallopian tube and extracted directly from there. So never went through the microenvironment or the chaotic microenvironment of the endometrium or the uterus, and never gone through birth. And so less genetic mutations and environmental mutations to accumulated. We also have the technology called GEMS, gene-engineered multi-sites, where we can put specific parking lots in our genome, and you can use CRISPR to insert whatever plasmid that you want, a CAR or any other plasmid, into these specific sites, and they will only show up there and nowhere else. So effectively solving the random insertion off-targeting issue. And we're trying to build this into our iPSC platform so that you have a true plug-and-play system. We have 47 patents to date, with a lot more coming, and this is our primary defensibility. This is the team. Myself, I've been working at this for 18 years. I also have a background in medical devices. CFO is an investment banker from New York, and Martina Molsbergen, our CBO, she's sitting up here. She's a lot of experience doing BD work in healthcare. And Siko Patma, our CSO, came out from UPenn, worked at Carl June's lab, and in 13 years at J&J. On the left, Jiaonan Li is my father. He's the one that discovered the stem cell platform. He's a KOL for OBGYN in Taiwan. And he did his MD in Japan and his PhD in the University of London, St. Bart's. Also in our SAB is Mahendra Rao, former director and a founding director for the Regenerative Medicine Center at the NIH. Ben Fryer is our main CRO. He's the company that primarily validated a lot of this work that came out of the university. And Peter Hudson and Bram Bout are product guys. Bram Bout famously was the lead designer developer for the Percy 6 line, and he is currently helping us turn our platform into a biomanufacturing platform. So in short, we have two primary platforms that we can offer to you. The main TSCs, as well as a TSC-derived iPSC. And for both of these platforms, you will get full FTO to use, and we will provide the regulatory support package to come with it, solving the CMC documentation issue. RUOs are available now, and the GMP is in progress. We hope to complete the GMP for the TSCs by June of this year, and fourth quarter for the TI PSCs. And subsequently, we'll file for DMF at the FDA. So we provide product market fit. We give you a platform with all the desired cell characteristics. We've de-risked the FTO and the CMC documentation issue. We can provide you with scale to reduce your individual dose cost. We can help you accelerate your product time to market and hopefully transform the current autologous service model to a allogeneic distribution model. And really our goal is to make CGTs available to all patients, affordable to all patients and available. It's a picks and shovels play. So We think that for every company that we license to, we can help them save 3 to 5 years of development time and probably $10 million in costs. And so that's our business strategy in short. So if you are interested in partnering with us and you're looking for an allogeneic cell line, come talk to me or any of my members. And I have this deck available to you. If you just find me and give me a business card, I will send it to you. That's great. So you can also take a picture or write down my email address if you don't want to come up and give me your card. So thank you very much.